Hi, my name is Mallory Palatucci. I am a PhD student rotating in Richard Cripps's lab, and we're studying muscle development and differentiation and disease. So what my project is based around is, can we use this new CRISPR-Cas system called prime editing in vivo and in non-dividing cells? In your regular CRISPR system, what you have is a Cas9 endonuclease that makes a double-stranded break, and we typically use homology-directed repair, where we provide a template that is incorporated after that in the single guide hybridizing to the DNA so that it has a template to repair itself. And this is very useful in cells that are dividing. But when cells aren't dividing, like you see in skeletal muscle cells or most muscle cells in model organisms like Drosophila, is that this homology-directed repair doesn't really work at a high efficiency because they don't have the division mechanism that goes on in normal cells. This new system called prime editing that uses a single guide RNA that already has a repair template attached to it that can hybridize to the DNA. It also uses a different type of Cas9 that only makes one nick in the DNA, so it's fused or mutated to be a nickase, and it's fused to reverse transcriptase. So what this allows the cell to do is repair itself without that mitosis, and we can use this system theoretically in non-dividing cells. So after it binds, the Cas9 will hybridize to the DNA and start editing. And then there's direct polymerization where this elongates and then it is able to edit the DNA. But the problem is now we have this 3' flap that's hanging out that needs to be incorporated. This will go through equilibrium, and in theory the cell system will prefer the non-mutated or correct version. And then the PAM sequence that is also in the normal template will be cleaved away because it's mutated out of the template that we've designed. And this will lead to a repaired or edited DNA so that you don't see the mutations in diseased muscle cells that we're trying to fix anymore. We tried this in 2 different genes using nonsense mutations that were easy to fix. So we used actin 88F and indirect flight muscles, and that's going to be in non-dividing cells. And we also used MEF2, the MEF2 gene, because we wanted to look at how this would work in undifferentiating cells that we're supposed to differentiate. And actin 88F wild type allows flies to fly because it's an indirect flight muscle, myofibrillar actin. And when you mutate this with a nonsense mutation, you get flightless flies. So what you should see when we use this prime editing system is restoration in flight. We can also look at this through IF and stain the indirect flight muscles to see that if it goes away once we mutate this, if once we rescue with prime editing, that IF is restored for the stain we're looking at. We also looked at MEF2, or myocyte enhancer factor 2, that's found in the pupae of developing Drosophila. And what MEF2 does is it allows the muscles in the pupae to actually develop. So in wild type, you see this nice staining, which is how we would validate this prime editing system, is look for a stain in the differentiated muscle cells once they start to develop. But in the mutant version, in the nonsense mutation, you won't see any differentiation at all, and it'll kind of just look like a big blob. So when we use the prime editing system to rescue this, even a little bit of rescue should be pretty noticeable because you'll see that skeletal muscle or the muscle in the Drosophila pupae start to come back with the rescue. With that, thank you for your time and have a wonderful evening.